Welcome to Ask the Expert. Today, we're very excited to have Dr. Steve Parker joining us, and his talk will be titled From Diabetes Genetic Variant to Single Cell State. What's the context? A very, very, very important question in type 1 diabetes and other autoimmune diseases. Just a quick bio. Steve Parker is the Associate Professor in Department of Computational Medicine and Bioinformatics, joint appointments in both the Departments of Human Genetics and Biostatistics at the University of Michigan. His career path includes a BS in biology, MS in molecular biology, and experience in industry, a startup biotech and large pharmaceutical companies, research at a large-scale genome sequencing center, PhD in bioinformatics and systems biology, and a postdoctoral training in genetics and functional genomics. He's contributed to completion of the human reference genome, identifying functional regulatory elements therein, and developing algorithms to interpret non-coding DNA. His research group studies the effects of genetic variation on chromatin architecture and transcriptional regulation at single cell resolution. The major goal of the lab is to generate mechanistic knowledge about how disease susceptibility is encoded in the non-coding portion of the genome, with a focus on complex metabolic diseases, including diabetes and related traits. Thank you so much for joining us. Really appreciate you taking the time out of your busy day to share your work with us.
Great, thanks, Monica. I really appreciate the invitation, and I'm looking forward to discussing with you and anyone else who's joined that has comments or feedback. I'd love to hear it throughout the talk. I think this is more of an informal kind of setting, so I think feel free to interrupt me whenever.
Yeah, exactly. People can drop questions in the chat, or they can unmute themselves or ask to be unmuted. We'll take questions as we go through.
Okay, sounds wonderful. So yeah, today we'll talk about diabetes genetic variation and how do we assign a context to a genetic predisposition. So I have a few, I made an outline here. I'll start with a little bit of the motivation about genetic risk and environmental exposures that underlie predisposition to type 1 diabetes or T1D. I'll tell you about a recent study design that we've implemented to characterize the molecular effects of T1D genetics under different contexts, including pro-inflammatory contexts. I'll show you some integrative analyses of single-cell multi-omics and genotyping data that we've generated now on 17 different donor samples. And then finally, I'll talk about some functional validation that we've done in collaboration with Shubing Chen's lab on prioritized molecular targets. So I'll start off and just say, type 1 diabetes, if you do a genome-wide association study, you see this Manhattan plot where the x-axis is position across genome and chromosome, and the y-axis is the strength of the p-value, and you see these big peaks that represent regions of predisposition. And there's been a few different GWASs published recently. This is from Cassie Robertson, Nature Genetics 2021, Joshua Chu from Kyle Galton's lab in Nature in 2021, and more recently, Michaela et al. This is out of Steve Rich's group. It's on medRxiv right now, just posted a week or two ago, trans ancestry GWAS. So your genome is kind of has all these different signals of genetic predisposition. The interesting thing is that if you look closer at any one of these peaks, it doesn't tend to overlap a gene itself. It lands in the non-coding space. So strongly suggests regulatory elements are involved in genetic predisposition, and identifying the context of those regulatory elements is therefore very important. So what cell types are those regulatory elements active in, and what are the target genes of those regulatory elements, and what are the contexts in which those regulatory elements might be active. So this is another way I like to think about complex traits in general, and T1D certainly is one. We have this kind of like layer of omics hierarchy where the bottom layer is your genome that encodes genetic predisposition. And then these genetic variants kind of percolate through all these omics layers, the epigenome, the transcriptome, proteome, cells and tissues. So ultimately they influence physiology and result in this complex trait, whether it's diabetes or waist-to-hip ratio or BMI or any other kind of genetically heterogeneous trait you want to think about. This is how we envision that they work. And I like to put or to emphasize that essentially your genome is invariant across all the cell types in your body. I know I put a little asterisk next to that because we're not talking about somatic variation here. We're talking about germline genetic variation, which is essentially the same across all your cell types. Every other layer in this omics stack is context specific, though. It can vary across cells and it can vary across the state of a particular cell. I like to put a gold star next to the epigenome because it is the most proximal molecular layer after the genetic variation itself. Like I said, remember, most of these genetic variations of association are non-coding space. So this genetic variant is likely influencing some sort of gene regulatory process, which then gets propagated onto the transcriptome and then further through all these omics layers that you can imagine until ultimately it influences physiology. So I'll talk a lot about epigenome profiling and in general regulatory element profiling. And the idea is that we want to integrate data modalities across these omics layers to build knowledge and glean mechanistic insights about how these genetic variations work.
Steve, can I just quickly ask, where would you fit the the microRNA profile? It's kind of is it like in between, you know?
Yeah, that's a great question. I mean, I guess it could, like, technically it's an RNA molecule, but you can see there's arrows connecting these things, and there's a bidirectional arrow between the transcriptome and the epigenome. And I think, microRNA is certainly, going from transcriptome to modulating the epigenome, right? It's one of those arrows that can go in either direction.
Feels like it might be in between that layer.
Exactly. And, I'm glad you brought that up because I didn't emphasize the directionality of these arrows. Essentially, all these layers are interconnected except your genome. Your genome is invariant across cell types, right? You're born with genetic variation. That genetic variation persists throughout your entire life. And so it's essentially an anchor of causality that we can use to infer direction from genetic variation. So yeah, but yeah, certainly microRNAs are somewhere in this region, I think.
Right. Thanks for setting that up.
Yeah. Thanks. Great question. So now to make this a little bit less nebulous and more focused on T1D, we know it's a product of both environmental stress and genetic predisposition in individuals. And of course, it all surrounds the important beta cell, right? The beta cell is doesn't live by itself. It's in this context. It has a microenvironment that surrounds it. It communicates with lots of other cell types within the islet microenvironment. It's exposed to immune cell signaling. This is all in the context of an individual's genetic predisposition and their personal levels of metabolic stress and even environmental exposures to viruses. So all of these things contribute to beta cell function and ultimately, I think, lead to T1D pathogenesis. So in order to investigate this, I want to introduce our interdisciplinary team. It's a collaboration between my group on the right, and this is a lot of work that initially was led by Ricardo Albunas, who was a PhD trainee in the lab. He's now a postdoc at WashU in St. Louis. And now this work is led, and actually most of the slides I'll show today are made by Cassie Robertson. Cassie Robertson, is a talented postdoctoral fellow. She came here from a PhD in Steve Rich's lab at UVA, who's a leader in T1D genetics and GWAS. Our talented collaborators are led by Shubing Chen at Weill Cornell and assisted by Angie Bo, Jerry Yuling, and Ziping Zhao. I'll show some results, some functional validation results, really nice experiments that Ziping has done later on in the talk. So our study design is to essentially take islets and look at them in a control environment, look at them when they're exposed to a cytokine pro-inflammatory cocktail, IL-1 beta and TNF alpha, and then also look at islets when they're exposed to Coxsackie B4 virus, where there's epidemiological data that indicates exposure to this particular virus can lead to T1D susceptibility. The study design is we do these exposure experiments, right? So islets get shipped to Shubeng's lab. She exposes them either to the cytokine cocktail or to the Coxsackie B4 virus. Then they freeze them, ship them down to us. We do omics profiling, lots of single cell, single nucleus multi-omics profiling that I'll go through. And importantly, our focus is to try to characterize regulatory elements. We also do genotyping on the samples. Then we do a lot of integrative computational analyses to integrate all the multi-omics data with genetic variation data, specifically from GWAS. And then finally, we collaborate, very tightly with Xubing Chen and her team. We nominate regulatory elements and target genes, and then Xubing Chen's lab does these really intricate and detailed experiments where they differentiate embryonic stem cells into beta-like cells and do CRISPR perturbations to figure out what is the effect when you perturb the target gene, what's the effect when you perturb the regulatory element that we've nominated. So I'll walk through kind of what we're seeing now, and I'll say that all of this data is currently unpublished. We're writing it up now and submitting it, so I'd love to hear feedback on this. So
just to qualify, Steve, that's all human cells, correct?
All human islet data. Yeah,
Great.
exactly. Yeah.
Thanks.
Yeah. So transcriptional regulation in cells. I'm going to show you first some cartoons of how we think about it and what these kind of single cell or single nucleus omics profiling modalities actually tell you. And I'll show you some real data. So here's a gene, here's a promoter where transcription factor is binding, some nucleosomes where the DNA is occluded, an enhancer where transcription factor is binding. Most folks nowadays are doing 3' end single cell or single nucleus RNA-seq. Almost all of it is 3'. This tells you, which genes are expressed, and it gives you the very 3' end of the gene. That's it. A really powerful assay is single nucleus ATAC-seq that we and others are doing. That tells you which promoters are open and accessible versus this promoter, for example, it's closed, and which enhancers also are open and accessible versus in this particular cell type or context, the enhancer is closed. Additionally, what we've been doing more recently is 5' end single nucleus RNA sequencing. So it tells you what are the transcription start sites within the nucleus, not based on cytoplasmic RNA at all. So it turns out if you have an enhancer element, it could be open in multiple cell context, but it may only be active in one cell context. And usually very active enhancers can be transcribed. And so we've kind of modified, this is not a conventional 10x prep, it's a slightly modified prep and modified sequencing and modified analysis, but with that, you can actually detect transcribed regulatory elements or enhancer RNAs here. So basically the idea is you want to identify a TSS or transcription start site that's associated not just with genes, but also with enhancer elements. And so finally, here's some real data. And this is at the PDX1 locus. PDX1 is a beta cell identity gene, right? So here's the beginning of the gene, and here's the 3' end of the gene over here on the right. I'm showing you now some real single nucleus data. This is ATAC-seq from beta cells, so we've already processed it. You can see a nice peak right at PDX1 like you'd expect. And upstream, you can see these peaks that correspond to known PDX1 enhancer elements. Here is the 3' single nucleus RNA profiling, and you can see clearly lots of reads piling up at the 3' end because obviously PDX1 is expressed in beta cells and islets. And then finally, here's the 5' end single nucleus RNA data, which I think is really, really exciting because you can clearly see the beginning of active transcription at the start of the gene, but these two open chromatin peaks identified by ATAC-seq, these known enhancer regions, you can see that they're actively transcribed, and you don't see that in the 3' end data. So the 5' end data, at least the way that we've kind of like tweaked this protocol, it allows you to identify active regulatory elements, not just any open chromatin peak. And so I think I'm going to skim over this really quickly because it's a little bit kind of a nuanced thing. But the way that we do our study design is because we've done genotyping on all the samples ahead of time, it allows us to mix within a batch samples from different donors. So here I'm showing you 4 different batches across the columns, and then the donors that are in each of these batches. So in batch 1, we have donor A, B, C, D, and E, and each of those donors has undergone, each of those samples, it reflects a different stimulatory condition of the donor, either CVV4 infection with the corresponding mock treatment or cytokine treatment with the corresponding mock or control treatment. And the thing that I love about this, and I get super geeked out about it, is that anytime you do omics experiments, there's these massive batch effects. Usually, if you look at PCA space, batch effect is like PC1 or 2 or very high in the PCs. Here, we're able to overcome batch effects because essentially we're randomizing our treatment conditions across the batches. And then we do our single nucleus omics profiling, and we're able to assign a nucleus to its corresponding donor by using the reads and comparing it to the already known genotypes. So the genetic multiplexing not only allows for these really nice, statistically well-controlled randomized block study designs, but it also allows us to superload or overload the 10x Chromium platform, and we just get lots more data that way, and it's much cheaper. So this is something I love to geek out about, but maybe it's a little bit in the weeds unless you're super aficionado.
No, I think it's really elegant.
It's one of the other projects that we work on is we're part of the Top Med, where we're analyzing about over 30,000 RNA-seq data sets. And the batch effects in that is just massive, right? So any omics data that I've ever looked at in my entire career, from graduate student till through postdoc and everything till now, it's always batch effects. So anything you can do in your study design to mitigate batch effects is a good thing.
Yeah, great protocol, and thanks for sharing.
Yeah, discover real biology. So here's the data that we've generated, and this isn't truly up to date, but for the purposes of the slides I'll share today, we've done single cell RNA-seq, single nucleus ATAC-seq. We've done multi-omic. That's where you get ATAC-seq and gene expression on the same nucleus. And then we've also done 5' single nucleus RNA-seq. Collectively, this represents almost 200,000 cells or nuclei across 17 different donors and 64 different samples. Because remember, we're doing this under basal or stimulatory context. So there's some donors get profiled multiple times under different contexts. Here's the UMAP, so the data looks good. We integrate across all these different modalities. We see beta cells, we see alpha cells, we see delta cells. We see what you'd expect, and the kind of marker expression looks good. Once we go and do the differential expression, so this is comparing control versus the cytokine cocktail. And we can look within a cell type. So in beta cells, we see 844 significantly differentially expressed genes, fewer in alpha, acinar, and ductal, and basically the amount of differential expression that we detect, I think, is mostly a reflection of statistical power from the representation of the cell type. So beta cells are a little bit more well represented than alpha cells, and certainly much higher represented than acinar cells in these islet preps. When you go and look at the GO-term enrichment, it's things that you would expect: interferon gamma response, interferon alpha response, TNF alpha signaling via NF kappa beta. So it looks like the experiment's working, is basically what I'm saying. We're getting reasonable and sensible results from these cytokine cocktail treatments. And just to show you, instead of such a broad view of the data, some of the top differentially expressed genes, here's 3 different genes that I'm showing you: the control condition in cytokines, the infection condition, and then the mock is the pair for the CVB4 infection. You can see IAPP is significantly lower expressed. JAK2 is significantly higher expressed, and PARP14 is also significantly higher expressed. So just to show the real data, and looks like we're seeing strong effects here. The thing that I want to get into and spend most amount of time on, though, is looking at T1D GWAS, right? I introduced earlier, there's over 100 T1D GWAS signals. And now we want to overlay this regulatory annotation data on top of the signals to try to figure out what are the relevant cell types and context, right? So here's an example where we just look at enrichment. We say, let's look at the ATAC-seq data. Are T1D GWAS signals enriched to overlap ATAC peaks and beta cells? And then here on the x-axis is the fold enrichment with a 95% confidence interval. So if the error bars are greater than 0, it's significantly enriched. So there's enrichment across cell types. Some of that is due to regulatory program sharing across the different cell types. But critically, you can see the beta cells are significantly enriched. This is just the ATAC peaks. Now, what if we look at that 5'N data and call regulatory elements using the 5' single nucleus RNA-seq? Here in red are the bars that correspond to the 5' RNA data. And you can see the error bars overlap with the attack data, but the point estimates uniformly are more enriched than the point estimates for the attack data. So suggests that transcribed enhancers may more precisely pinpoint active regulatory elements, not just the attack data. And I'm going to now go and show you some specific examples of this. But before I do, I want to show you the big picture. Here is a heat map where the x-axis are the 136 currently published T1D GWAS signals. The y-axis represents cell type. And what we did, Ricardo came up with this cool analysis approach where he took the genetic fine mapping data and integrated that with the chromatin accessibility data, and then created this kind of heat map representation based on per cell type accessibility on the genetic association credible sets. And where it's red, that cell type looks like it if it has very high accessibility. Where it's blue, it's low accessibility. So over here on the right, you can see these T1D GWAS signals look like they're very, very high in immune cells, so not surprising. Whereas over on the left, you can see these T1D GWAS signals look like they're highly active in beta cells, for example. So already we can see this potential influence of immune cell types on the beta cell, effects directly on beta cells, and also potentially communication within the islet microenvironment. You can see alpha, delta, gamma, all these other cell types look like they are have very context-specific association with T1D GWAS and influencing potentially beta cells. So understanding communication across the cell types would be important moving forward. So now I want to go and show an example of prioritizing causal variants and genes at a particular T1D GWAS locus. So again, on the bottom is a Manhattan plot. If you zoom in and look at one peak, you tend to see something like this with lots of genome-wide significant variants, lots of them in high linkage disequilibrium with each other, and hard to know what is the causal variant there, and importantly, what's the target gene, because that's where we want to go for therapeutic effects, right? So here's an example at the GLIS3 locus, where you can see the gene GLIS3. Here's the T1D GWAS credible set. You can see all these variants are in the credible set. None of them overlap exons. These are all intronic to GLIS3. And then here now is our overlaid data. You can see this variant looks very interesting because it overlaps the summit of an attack-seek peak that's in beta cells, alpha cells, and other endocrine islet cell types. What's really interesting is if we look at this in closer detail using the 5' end data. So here is that SNP again. Here's the attack data in beta cells, the attack data in alpha cells. But here's the 5' end RNA data. This, open chromatin peak enhancer element is highly transcribed in beta cells, not in alpha cells at all. So this is a really good example, I think, of, you have chromatin accessibility in both alpha and beta cells, but it looks like the regulatory element is only active or highly active in beta cells, not in alpha cells. So this is where context is critically important. It turns out this same variant was, in a small part in a previous publication. Well, actually, sorry, the cartoon looks like this. So beta cells, alpha cells, both of those cell types have open accessible chromatin, but only the beta cell enhancer is highly active, and you see the transcribed RNA coming off of it. So if you look at this Miguel Escalada Nature Genetics paper from a few years ago, 2019, they actually looked at this variant and they did CRISPR studies. They did, importantly, just Cas9 only, nothing, controls outside the region, and then controls on the enhancer deletion. And the enhancer is this enhancer that we identified right here. When they delete that enhancer, GLIS3 expression goes down. So GLIS3 looks like the target gene, and it looks like beta cell. Oh, and they did this in endo-C cells, so a beta cell model. And it looks like beta cells are the potentially causal cell type. So I'll tell you one more story before I close. This is at a different locus. This is at the RAS-GRP1 locus, where again, Ricardo initially led this work and Cassie has now picked it up. Here's the credible sets. Here's the beta cell attack data, the alpha cell attack data, and then the 5' end RNA data. So active, clearly active regulatory element in beta cells, and a little bit active in alpha cells too. This T1D GWAS signal is also a signal of association for Crohn's disease, for hypothyroidism, and for serum 25-hydroxyvitamin D. So other genome-wide significant results for this variant. What we did is we communicated back with Ximing Chen's group, and Ziping Zhao is a talented postdoc in that group who's leading some of the functional validation experiments where he's taking, they have an GFP, an insulin GFP labeled embryonic stem cell line, so they can track beta-like cells after the differentiation. So they create isogenic embryonic stem cell lines with CRISPR perturbation. Then they differentiate these ESCs to beta-like cells. And then they do a bunch of kind of experimental procedures to evaluate the effects on function and on survival. So RAS-GRP1, here's what happens when you do you do CRISPR to delete the gene itself. Here's the after differentiation. The y-axis represents the percentage of GFP positive, so insulin cells. Wild type RAS-GRP1 homozygous deletion, significantly fewer beta-like cells. Also, there's more annexin V positive cells. So this is a marker of apoptosis. So wild type versus RAS-GRP1 deletion, significantly higher signatures of apoptosis. So looks like loss of RAS-GRP1 impairs beta cell differentiation and causes beta cell death. That's the gene itself. What about the regulatory element? So they went back to this regulatory element, did a regulatory element deletion. And the first question is this regulatory element corresponding to RAS-GRP1? So it is. So here's the expression level of RAS-GRP1 in wild type and in the RAS-GRP1 delta, which corresponds to the regulatory element deletion. So gene expression goes down when you delete this regulatory element. So it looks like the non-coding regulatory element does in fact regulate RAS-GRP1. When you delete the regulatory element itself, here's again the percentage of GFP positive beta-like cells after the differentiation protocol, wild type, and RAS-GRP1 regulatory element CRISPR deletion. So fewer beta-like cells, so impaired differentiation phenotype with regulatory element deletion, not gene deletion. And just to put that back in context, here's when you delete the gene itself, you get kind of a pretty big effect. When you delete only the regulatory element, you still get a significant effect, but it's not as big of an effect as when you delete the entire gene, right? Presumably, there's other regulatory elements and things influencing expression of this gene, not just this one that was deleted. So cool kind of validation experiment. And I guess I'll just summarize and say that there's cis regulatory elements in multiple human islet cell types that are enriched for T1D associated genetic variation. We think this signature of transcribed regulatory elements that you can see with the 5' end single nucleus RNA data may more precisely pinpoint active regulatory regions underlying T1D associations. We've done computational integration with T1D GWAS credible sets, and it suggests beta cell target context mechanisms in multiple loci, including GLIS3 and RAS-GRP1. And right now we're doing functional validation on multiple different, nominated causal genes, regulatory elements, and at variants using this ESC beta cell differentiation model. I guess I'll wrap up, and I want to acknowledge a lot of folks. This is work that originally was led by Ricardo, but now he's a postdoc at WashU. Cassie has come on to lead this and has done a fantastic job. Rushi and Peter have also contributed a lot to this. Nandini Manikam is a former lab tech in my group who generated a fair bit of this data. Xubing Chen's lab has contributed massively to this. She's incredibly talented, and so are all the trainees in her lab, including Ziping. We collaborate with a fair number of other groups at Michigan, Francis Collins at NIH, where I did my postdoc, Marcelo Bersova now powers at Vanderbilt, and other folks across the world. We're members of MOTAPAC, of the Accelerating Medicines Partnership for Common Metabolic Diseases, Human Islet Research Network. This is the Top Med logo, and we are generously supported by a few different places. So thanks to those folks too. And finally, I always like to end the talk with thanking the organ donors. Without those precious samples, none of this research and knowledge that's being built from it would ever be possible. So thanks again for the invitation. I'm happy to chat about anything.
Yeah, fantastic, Steve. Thank you so much for this, sharing this beautiful work, and just kudos for all these, strong collaborations across some of the top players in this field, for sure. And I wanted to open it up to the audience. If you'd like to drop something in the chat or just unmute yourself, you feel free to. I'm just looking to see if anyone is unmuting themselves. I wanted to ask you this question. You know, have you thought, this is a sort of a large question, but I mean, have you thought about why some individuals, right, may have some more highly reactive regulatory elements in beta cells? Or is that even the case? Like, do some of these people have more highly reactive regulatory elements, and that's why the beta cells, respond to cytokine assault or viral assault? Or, is it just that, every across the board human beta cells respond by using their regulatory elements, and then it's just a contextual issue of the immune response? What are your thoughts? And it's a huge question, but
Yeah, no, that's a really great question. I think I haven't seen data to address this at the single cell level in islets yet, but I'm kind of maybe kicking myself a little bit for not sharing. We have some skeletal muscle data that's much larger in scale than the islet data that we have, and maybe I could share, if you want, I could share a plot that kind of illustrates this.
yeah,
But the
that'd be
idea
great.
is yes.
No,
Okay, sure. But like just conceptually, the idea is yes, personalized genetic variation can have a profound influence on regulatory element sensitivity, right? So depending on which allele you have, your chromatin could, be basically wide open or almost completely closed.
right.
And that kind of sets the stage for, all right, if you have open chromatin and then you experience a certain environmental context and there's a binding site for that context there, you're going to get a big response. And so
Mhm.
let me, I'll stop sharing this screen and I'll share you, this is more unpublished data. It's all in skeletal muscle, but it's going to I think directly address what you were just saying. Okay, so we have this manuscript we're working on. Here's one of the figures. And this is chromatin accessibility, but it's partitioned by genotype. So in red are individuals that are homozygous T, in orange are individuals that are heterozygous CT, and in black are individuals that are homozygous C. So
Mhm.
depending on your genotype, there is a massive change. This is what's called a chromatin accessibility quantitative trait locus, or a CAQTL, where genetic variation influences chromatin accessibility. Huge effect size right here, right? And there's at least one study published. Michael Stitzel has this beautiful work in bulk islets. So I'm not aware of any work in single cell or single nucleus islets yet. The bulk islet work clearly shows this is going to happen. Is just kind of conceptually, it's definitely going to happen. But once our group and other groups I know are working towards this to generate larger numbers in islets, you're going to see similar kinds of things. And my prediction is you're going to see massive genetic effects on regulatory element usage, specifically at T1D GWAS signals that basically are predisposing these people to kind of different environmental contexts.
Yeah, and then you can go about maybe teasing out those environmental insults and what's happening, right? Like, okay, is it Coxsackie-B? Is it some kind of SARS-CoV-2? Is it, what is
Yep,
it?
exactly.
So
Yeah.
that is so beautiful. It's really interesting. I wondered, in terms of like, what would be a way to address this once you find it? Once you find those who are, have tons of open chromatin, is there any other, disease that really, has this at its baseline, more open chromatin and resulting pathology?
Yeah. So I think basically most common complex diseases are going to work like this, right? And the risk allele that could result in open chromatin, it could result in closed chromatin, but the idea is genetic variation is kind of fine-tuning regulatory element landscape. And then on top of that, you get these environmental perturbations that operate and are, the effects of those are propagated based on your kind of like genetic pre-configuration. In terms of what do you do with it, I think,
How do we mitigate these the risk?
yeah, I guess the first thing is like understanding. Right now, GWAS is wonderful, but it doesn't tell you anything about context. It's completely agnostic to what's the underlying mechanism. You just
Yeah.
know there's genetic association. So now I think the kind of first wave of influence is going to be understanding what are the cell types, what are the regulatory elements, and what are the target genes, right? Once you start understanding those, you can start building network models to understand, okay, here's the pathway that looks like it's systematically disrupted by genetic variation. And it's probably going to turn out that every person is a little bit unique. Like one person, maybe they have genetic predisposition that's loaded up in their immune system, but their beta cells are fine. Another person, maybe their genetic predisposition is loaded up in the beta cells, but their immune system's totally fine. And you want to treat each of those individuals a little bit differently based on their personalized genetic predisposition. So going from, a genetic signature predisposition to kind of tissue specific pathophysiology, I think is kind of the next step of where the field is going.
The march towards personalized medicine.
Yeah, that's the goal, I think.
Good. Well, I like it. I always kind of go back to this example, but even 20 years ago, breast cancer was sort of like one and done, and now it's much more personalized. So why can't complex diseases like type 1 also go there? There's no reason.
Exactly.
So
Exactly.
thank you. Last call for questions. Anyone else? See, City of Hope is here and other people. All right. Well, if you have any questions, reach out to Steve directly. Are you looking for any postdocs?
We are, yeah. Sorry, that was in my last slide and I forgot to say it, but we're always looking for postdocs. So please, if anyone's on and interested, shoot me an email, and Monica and your network, if you know of anyone, please
Will
send them
do.
our way. We'd love to meet folks.
We will. Yeah, it looks like there's a lot of work to be done over there in Michigan. It's a great place to be. Very vibrant area right now for type 1 with the Caswell Diabetes Center in full force. So thank you again. Fantastic. We'll be looking for your next papers, and we really enjoyed speaking with you today. Have a great rest of your day.
Wonderful. Thank you so much, Monica. Congratulations on everything you've done with Sugar Science, and thanks again. Take care.
Thank you. Bye-bye.